Hello, I'm Sophie Rovner from the American Chemical Society. Welcome to this news briefing from the ACS Fall 2020 Virtual Meeting and Expo. We're joined today by Dr. Scott Eagon from California Polytechnic State University. He's studying targeting iron uptake to create a new class of antibiotics against UTIs. Dr. Eagon.
Thank you for the invitation. I'm really excited to speak to you today. So my group at Cal Poly, we're a medicinal chemistry group. We're really interested in targeting infectious diseases with new small molecules. In this particular project, we're looking at urinary tract infections. So this is a big issue in the United States, around 11 million cases, costs nearly $3 billion annually to treat. And about half of all females in the United States will experience at least one UTI in their lifetime, with almost a third of those experiencing reinfection. Now, the primary pathogen that's involved in these UTIs are uropathogenic Escherichia coli, or E. coli. If you go to the next slide, please. Thank you. So when the E. coli invade the human urinary tract, they find themselves in a kind of an unusual environment in that there is not a lot of iron around. And these UPEC strains, just like humans, they need iron to survive. So when they're in this very iron-poor environment, they have to go scavenging for iron. And the way that the E. coli do this is they'll release molecules called siderophores. And in the slide at the top there, you see a little orange triangle, that's a siderophore. The siderophore is basically this little molecular net that when it finds iron floating around, it grabs onto it and complexes with it, holds it very tightly and won't let it go. Once the siderophores have grabbed the iron, then the bacteria will pull that into what's called the tone B dependent transporter, that TBDT, right? It's that tube at the top. And that's basically what it is. It's this big beta barrel tube that can allow the siderophore and the iron to come in. And once it's there, then the critical part is transporting it through the membrane. And what does that is called the tone B complex. And that's that little three cluster of proteins there in blue in the middle, right? You'll see tone B, XBB, and XBD. The tone B complex it's basically the motor. It gives the proton motive force to pull the siderophore through the membrane into the periplasmic space, excuse me. And then now the bacteria can use that to survive, right? So my collaborators, which is Harry Mobley at the University of Michigan and Alejandro Yep, also here at Cal Poly, they wanted to look for small molecules which could target that tone B and try to shut down that transport of iron. So they screened about 150,000 compounds and were able to identify 2 molecules which specifically inhibit tone B. And the really exciting thing about that is by going after the iron transport, you're only killing bacteria that are in the urinary tract, right? And you're leaving other bacteria alone, right? A lot of antibiotics which are used now are broad spectrum, meaning that they are going to kill bacteria both in your gut and in your urinary tract. And E. coli is perfectly fine to have in your gut and beneficial, right? So by going after tone B, which is only utilized in the urinary tract in these iron-poor environments, you can selectively just kill the bacteria that are in your urinary tract and leave the beneficial ones alone. So there's still a lot more to be done, right? The 2 molecules that we've identified are not potent enough yet for humans. And so my group, our goal now is to take these 2 molecules and see if we can increase the potency, try to figure out how to make them better and eventually start advancing them through preclinical trials and hopefully in the future identify something for clinical trials. But that's pretty much a summary of what we're doing. I'd be happy to take questions and answer them as best as I can.
All right, thank you. So at this point, we would invite questions from viewers. And if you have one, please state your name and affiliation in the chat window when you type in your question. So I'll get us started with this one. How common is it for the bacteria that cause UTIs to become resistant to antibiotics? And is this a widespread problem?
Yeah, it's the bacteria that get in the UTI are just like any other bacterial infections. Like they are all developing antibiotic resistance. And I don't have the numbers off the top of my head, but I do know that the resistance to the classes we have now are increasing. UTIs are commonly treated with a fluoroquinoline or the amoxicillin classes, and those more and more resistant strains are coming out. So another bonus of this is that no other no clinical candidate now or anything in the clinic works this way, and so it would definitely be a boon for any kind of an infection in the urinary tract that had resistance against current treatments.
And what gave you the idea to target iron uptake for a new class of antibiotics?
I have to give all of that credit to my collaborators. So Dr. Alejandra Yep and I both arrived at Cal Poly at the same time in 2014. Her background is in microbiology and biochemistry. I'm a medicinal chemist. We were new faculty. We're like, what are you doing? What are you doing? And we tried to come across this, a collaboration. And when she told me about this work she'd been doing with Harry Mobley at University of Michigan, I said, well, that's fantastic. Let's work together. So it was entirely their idea to target this. And then I, of course, recognized that as, oh, this is different than anything else in the clinic. Let's go after it.
And why do bacteria need iron? How do they use it?
Well, you're asking a chemist a microbacterial question, so I know it's essential to their metabolism. I don't know exactly what they use it for. Yeah, as a med chemist, I just need to know how to break the machinery. I don't need to know how it works. So that's a bonus of my field. So yeah, I'm sorry, I don't know the answer to that.
All right, no worries. And why did you decide to focus on the two tone B inhibitors identified in your screen?
So those were the only two that they identified. So, the last time we looked, those are the only inhibitors in the literature that target tone B at all. And so it's all we have to work for. I mean, certainly they're not, yeah, it would be nice if there were more potent things out there as a better starting point, but these are the only two that are out there. And it's such a unique mechanism that, my group and I, we really wanted to tackle this. Like this is, as far as I know, no one else is working on this. So we hope that it has the promise of a lot of impact if we can find a way to make it potent enough for humans.
And we have two questions actually from Kath O'Driscoll, Society of Chemical Industry. First question from her, could these molecules interfere with beneficial iron uptake in humans?
So my collaborators, Alejandra and Harry, they did cross-screen to make sure that these molecules aren't actually siderophores themselves, that they're not actually stripping iron themselves. So to the best of our knowledge, no, they should not interfere with any kind of human uptake, and they're not complexing the iron itself. But we have not tried these in human cells yet, so I can't answer that for sure. But the current working hypothesis is no, it's not actually interfering with the iron itself. It's interfering with the machinery that's only present in the bacteria. So humans don't have tone B. So we don't believe that it will cause any problems with human normal iron uptake.
And she says a related question, are there any other downsides to targeting tone B?
Again, since there is no human equivalent, there's nothing like this in humans, there's nothing that we know of. And that's another thing that made it a really attractive target for us, that there's no human equivalent. So yeah, I certainly can't guarantee there aren't going to be any toxic effects. We'll have to test it in human cells first. But currently, no, we don't think so. We're going after something that's only in the bacteria, and we don't think so. But we'll see. We'll see.
Do you know what aspect of iron uptake the other 14 compounds you identified target?
So with those other molecules, no, there's a lot of machinery in there. You have both an outer membrane and inner membrane. So our collaborators were only trying to target tone B, and some of the other molecules they looked at were complexing iron itself, or they were killing the bacteria through some other mechanism. And so we were, or I should say, they were laser focused on just finding tone B. And so those are the two that we went with. So now we don't know. They didn't investigate further on those.
Okay. So from that answer, it sounds like the other 14 compounds then are not going to be pursued for a slightly different direction?
Not by us at this time. I'm not sure if the Mobley lab is trying to figure out how those other active compounds are working. It was very complicated. They had there's lots of associated machinery in that picture you can see, and so there's many things that could be interacting with. Whether or not they're able to figure that out, that's quite a challenge. We just took these two compounds because they were the most clear to us. But it certainly would be possible with additional studies, they may identify more and figure out what's going on. And we would certainly, we think the UTIs are a big problem, and we would take any path that we thought would be promising.
Over time, could the bacteria evolve mutations in the TONB system that would make them resistant to this type of inhibitor?
Yeah, that's certainly possible. One of the advantages of going after TONB is it's highly conserved and it is critical to the survival. So even if it could evolve resistance, which it certainly could, it would pay a price for that, a fitness price. Yeah, I don't think it's impossible for it to do so, but one of the best ways to combat evolving resistance is to cycle different classes of drugs. So have different drugs that attack it different ways. So if it becomes resistant to one class of medication, then you switch over to another class. And so the more kind of bullets we have in that magazine, the more different ways we can cycle to attack it. It would prevent it from it can't evolve resistance to everything. It's going to have to sacrifice something. So it certainly is possible, but we would not expect any resistance to be present now because it's never been attacked this way.
All right, let me see. By how much do you think you need to increase potency to make the compounds useful?
Yeah,
and
right
now sorry, go ahead.
No, go ahead. I'm sorry.
Second part is, do the compounds require specific properties like being able to cross the bacterial cell walls?
Yeah, so for the first part, yeah, we're going to have to increase by several orders of magnitude. So right now they're in the single micromolar, which is considered pretty weak. And frankly, for a lot of compounds, if I were looking at them and somebody had something at that level, I probably wouldn't even consider it. But the reason that we're choosing this one is because their mechanism of action is so unique. The way they're killing it is so unique. So it's definitely a high risk project, perfect for an academic lab like mine. But the rewards are really great, right? You could develop a whole new class. And then, yeah, of course, because we're targeting something that's in the inner membrane, it's going to have to diffuse through membranes. Most of the drugs that we make in my lab, they're designed to be taken orally, so they already have to diffuse through human membranes. And that cell membranes are not that different from the outer membrane of bacteria. So yes, we're going to try to design them so they're permeable. Ideally, you should just be able to take something by mouth. We don't want to have to apply something locally. But yeah, that's all part of the drug design process.
What types of modifications did you make to the hydroxyquinoline scaffold, and why did you decide to make those modifications?
Yeah, so the real challenging thing with TONB is that there are no full crystal structures of this, and it's not even understood how it works. So we're looking for a needle in a haystack, and we're doing it in the dark. So we really don't know what we're doing, and that's why it's called research, right? Again, another perfect project for an academic lab. Right now, we're basically taking the original molecules that our collaborators have discovered, and we're trying to chop off certain parts over here, or chop this off, and we're trying to figure out, well, which parts are important? Because if we chop something off and it doesn't change, well, it tells us it wasn't doing anything. But if it loses activity and becomes even worse, we're like, oh, that's the important one. So we're trying to figure out what are the critical parts of the drug. And then once we do that, we're basically going to use a set of, there's a few sets of standard functional groups we can add to drugs in medicinal chemistry, a little more polar, a little non-polar, and try to figure out, what is good and what is bad. And if we can figure out, oh, polar over here is good, and then a greasy thing over here is good, ideally, then you can assemble all that into something which is overall potent. But again, a lot of it is just guesswork, trying to figure out, what works, what doesn't work, how can I combine all the best things together. It's going to keep us busy for a while.
So it sounds like it might still be too early for you to have tested any of the modified inhibitors in cells or animals. Is that right?
Yeah. Right now, we're just building up the synthetic library. My students have about half a dozen compounds, but it doesn't really make sense to screen things one at a time. You really need at least a dozen, or ideally 2 dozen compounds, so that you can get this whole sweep and you can try to rationalize, oh, what's working, what are the patterns here. So yeah, my colleague Alejandra has the ability to screen them now, but we're waiting until we get a big enough collection to put them through.
And could these inhibitors be used to treat conditions other than UTIs?
So the urinary tract is the only one I'm aware of where it's an iron-limiting environment that you could use it. But there may be other applications I haven't not aware of them. Yeah, we were primarily just focusing on UTI themselves and taking advantage of this iron and poor iron environment to go after tone B, hopefully. But yeah, it's kind of beyond the scope of the current project.
Okay. All right. So lastly, I would just ask you to summarize a take-home message you'd like viewers to take away with them from your work.
Yeah, well, I guess the real take-home is we're just trying a real high-risk project to see if we can make a new class of antibiotics. And if we can, we really hope to help a lot of people. All right. Well, thank you so much. The archived version of this session will soon be posted at www.acs.org/fall2020 press conferences. Please join us for our next press conference tomorrow at 9 a.m. Thank you.